For many years, astrocytes, the star-shaped cells in the brain, were thought to be nothing more than supportive sidekicks to neurons. But as scientists have peered deeper into the mysteries of the brain, a new story has emerged. Astrocytes are not just passive helpers, but active players in brain health and disease. In Alzheimer's disease, the brain becomes littered with sticky clumps called amyloid beta plaques. Astrocytes, sensing trouble, spring into action. They try to clean up these plaques, swallowing them through a process called autophagy and breaking them down using a metabolic pathway known as the urea cycle. At first glance, this seems heroic. But as the astrocytes work overtime to clear the debris, something goes wrong. The breakdown of amyloid beta by astrocytes sets off a harmful chain reaction. As they process the plaques, astrocytes begin to produce too much GABA, a chemical messenger that normally calms brain activity. In excess, GABA dampens the brain's signals, leading to memory problems. At the same time, this cleanup process generates hydrogen peroxide, a toxic molecule that damages neurons and accelerates brain degeneration. Curious about what drives this overproduction of GABA, a team of researchers set out to find the key enzymes responsible. Using a combination of molecular detective work, high-powered microscopes, and electrical recordings from brain cells, they uncovered two main culprits, SIRT2 and ALDH1A1. These enzymes were found to be especially active in the brains of mice with Alzheimer's and in human brains affected by the disease. When the researchers blocked the action of SIRT2 in the brains of Alzheimer's mice, something remarkable happened. The mice showed a partial recovery in their memory, and the flood of GABA slowed. However, the improvement was limited to short-term working memory. Other types of memory remained impaired. This result was both promising and puzzling, hinting at the complex web of changes that Alzheimer's brings to the brain. Digging deeper, the scientists discovered that SIRT2 acts at the final step of GABA production, while hydrogen peroxide is produced earlier in the process. Even when SIRT2 was blocked and GABA levels dropped, hydrogen peroxide continued to be released, suggesting that some forms of brain damage might persist despite the reduction in GABA. This discovery marked a turning point. By pinpointing SIRT2 and ALDH1A1 as the enzymes responsible for GABA overproduction, researchers realized they could now selectively block GABA without interfering with hydrogen peroxide. This is a crucial advance because it allows scientists to untangle the separate effects of these two harmful byproducts and better understand their roles in Alzheimer's progression. Previously, treatments targeted an enzyme called MAOB, which blocked both GABA and hydrogen peroxide. Now, with the identification of SIRT2 and ALDH1A1 as downstream players, it's possible to design more precise therapies that target only GABA, leaving hydrogen peroxide untouched for further study. While SIRT2 may not be the ultimate target for new drugs, since blocking it doesn't stop all the damage, this research opens the door to smarter, more targeted strategies for controlling the harmful changes in astrocytes during Alzheimer's disease. The story of astrocytes is no longer one of silent support, but of active involvement in the fate of the brain, offering new hope for understanding and treating this devastating illness.